Hi, it's Miss Lee here. We are going to look at cellular respiration today. Now, for our learning objectives, we're going to look at the two types of cellular respiration. One is called aerobic respiration, and the other one is called anaerobic respiration. You have to know the equation for aerobic respiration, and we will look at the structure of the mitochondria. And that's going to show you where each step of the aerobic respiration happens. There are three major steps, and you'll need to know what they are. Now, today, we're going to specifically look at the very first one, the very first step, which is called glycolysis. Cellular respiration is a metabolic pathway, meaning that it has multiple steps or multiple chemical reaction that uses biological fuel to produce adenosine triphosphate, ATP. Now, if you remember, ATP is the universal energy providing thing or chemical molecule inside a cell. Every single cell on Earth uses this. Biological fuel, specifically for animals, for us, it's glucose, all right? So cellular respiration means breaking down glucose to produce ATP. There are two major types of cellular respiration, aerobic and anaerobic. The Latin root of these two words is the little aero thing. Now, aero in Latin means air. So if we look at aerobic respiration, that means it's the one that uses air, and this one is the one that doesn't use air. Now, inside the air, specifically speaking, we're interested in oxygen. Oops, I'm spelling oxygen wrong. So oxygen is required for aerobic respiration, and anaerobic respiration doesn't need oxygen. Now, human do both of them, which means humans have a way to produce ATP without using oxygen at all. And you might wonder, if we don't need oxygen to produce ATP, why don't we just use this? Why can't we just go around without breathing? Why do we die if we do not get air in 3 minutes? Well, we're going to get to that in 2 lessons' time. Now, first, let's look at the diagram, all right? This is a very uglily drawn diagram, I know, but it gives you all the hint as to what is inside an aerobic respiration. So on the left-hand side of the arrow, which is where your inputs are, you have number 1, the air that goes in, and that is oxygen. Oops, that's too thick. Now, oxygen is one of the input. The other input is the chicken drumstick, number 2. And chicken drumstick, that just means glucose, all right? It means food. And as an output, the first output you have breathing out is your carbon dioxide. Carbon dioxide. The other one would be this right here. Now, P has uric acid, it has electrolytes, it has water. So now, to be honest, water is the major output. The rest of them, they don't exactly get produced in cellular respiration. Rather, it's a waste product from other processes. So we're just going to put down water here. And then the third output, which is our ultimate goal, that is our ATP. Now, if we turn the word equation to chemical reaction, you're going to get O2 plus, if you remember, glucose is C6H12O6. And we have carbon dioxide, water, and ATP as an output. So that's our chemical equation for an aerobic respiration. Now here's a picture of the mitochondria if we zoom in and look at a cross section. What I want you to do is, without looking up without looking it up on Google, I want you to try and label the diagram. I will give you a hint though. This is the definition for a couple of those. So in your notes, I want you to pause the video and try and label the diagram for me. Okay, hopefully these are the answers that you have. The outer membrane and the inner membrane really is not difficult. I think it's common sense. Now the matrix, we were referring to the space that is inside the inner membrane. And then the cristae are the ones that are the folds created by the inner membrane. And the space between the inner and outer membrane, we call it intermembrane space. Now, the reason why I'm teaching you this is because aerobic respiration have 3 major steps, and they happen at different places. So the first one right here is glycolysis. It happens in the cytoplasm. And the second 2 steps happens in the mitochondria, but they happen at different places of the mitochondria. So that's why I was teaching you that. I want you to use the diagram and try and fill in the blanks. I'm not going to teach you that because the diagram really does a good job. So interpret it and fill in the blanks, please. Okay, see if you got the answers correct. Aerobic respiration can be broken down into 3 major steps. You have glycolysis, that breaks down glucose into 2 units of pyruvate. And you can see that here. And then after that step, you have the Krebs cycle, and it happens in a mitochondria. During this process, the pyruvate goes into a series of chemical reaction. It gets modified, and during that process, we produce 2 cofactors, NADH and FADH2, and that is what we're really interested in. These 2 cofactors are then going to go into the 3rd step, where you have an electron transport chain, and they will utilize NADH and FADH to produce ATP. Now this process, later on when we look at it in detail, it may remind you of some of the stuff that we did in photosynthesis. Okay, so here you go. Here is glycolysis. Glycolysis, if you remember metabolic pathways, uses arrows to represent chemical reactions. So here's the map for you. And I trust that now you have the ability to interpret it. I want you to fill in the blanks for me for this first part. Okay, so the name of the initial substrate is right here, glucose. The name of the final product, pyruvate. Now the number of carbons in glucose, I hope that you would know because you call it C6H12O6, meaning that it must have 6 C's, 6 carbons. And the number of pyruvate, if you're splitting 1 glucose into 2 pyruvate, and they're identical stuff, which means you divide 6 by 2, and you have 3 carbons in each of them. The enzyme that splits the carbon ring into 2 identical precursors is called aldolase, right here. Hopefully you got those correct. Now, if we look at NAD plus and NADH, it is a cofactor, or you can also call it a coenzyme, and it acts as a carrier molecule, so it can carry things for you. NAD plus is a molecule that is able to carry, or has the ability to carry, 2 protons and 2 electrons. Remember, hydrogen is made up of 1 proton and 1 electron. If you cut the electron off, it just leaves the proton behind. So that's why H plus without the electrons is just protons. And here are the electrons. So NAD plus, it is considered an oxidizing agent. An oxidizing agent just means that it is something that has the ability to accept electrons. All right, so that's what we call an oxidizing agent. Now, for things that has the ability to donate electrons, we call them reducing agent. So this is your oxidizing agent. This is your reducing agent. So oxidizing agent do not have electrons sitting in their car already. They're waiting to accept electrons. So if you add the electrons to them, you can turn it into a fully loaded car, and that is a reducing agent. It has the ability to be reduced. All right. And so this molecule right here is important because later on, you're going to use this molecule in the third step. It can carry a lot of electrons, all right, if you get a whole bunch of them. And those electrons can be donated in the third step. And then you get that flow of electrons, which is you get electricity, electrical energy going. And that can be used to produce ATP in the third step. Now, it doesn't mean that we don't produce any ATP in glycolysis, the first step. In glycolysis, if you look at the net ATP yield, which means in total how much is being produced, you can see that at the beginning we have 2 investments of ATP. So we're investing 2 ATPs. However, after the initiation step, we get 1, 2, 3, 4 units of ATP being produced. So the net yield, if we look at this, ATP yield is 4, and then ATP invested is 2. If you do, sorry, I don't know why I wrote that. It should be a deduction, a subtraction here. There we go. So 4 minus 2, the net yield is 2. So you're creating 2 units of ATP for use in this glycolysis step. It is not a lot, so therefore it is not very efficient. However, in the very third step, the third step will produce 34 to 38 ATPs with 1 round of chemical reactions, and that's way more efficient. So the hard work is really being done in the third step. The major purpose of the first glycolysis step is to get these produced so that they can be used in the third step. Okay, a little summary. I would hope that you will work this out yourself, which is what we did in class. But if you're watching this from home, here you go. 2 units of ATP has been invested right here. And then we have 4 units of ADP invested as well. So they're here. And then we have 2 NAD pluses going in, which is right here. As an output from the ATP, you have 2 units of ADP. The ADP turns into 4 ATPs. The 2 NAD pluses turns into 2 NADH. And then right here you have a water molecule produced. All right, so 2 water molecules, and then the final product are the 2 units of pyruvate, and that's your summary. All right, CK12 has been posted as homework, so have a read, try to practice questions, and goodbye.